Hi, I'm Francesca Chen, and I am your host for Next Breakthrough, the Biomedical Innovation Brainstorm podcast. In this episode, our very first one, in fact, I interviewed Dr. Kelly McNagny. Dr. McNagny is a principal investigator at the School for Biomedical Engineering at the University of British Columbia in Vancouver, BC, Canada. His team of biomedical researchers are interested in hematopoietic stem cell biology. Throughout this episode, our main discussion was on a specific type of recently discovered immune cells called innate lymphoid cells. We discussed their role in inflammation, their relationship with bacteria and disease, such as Crohn's disease, and the next generation therapies currently being explored to regulate and control their function. Towards the end, we talked about the future of research in this area, the type of innovation that could deal with the problems we discussed earlier on, and the type of mindset needed to continue to drive this innovation forwards. Finally, Dr. McNagny gave us a teaser on a potential following episode with him about the novel cancer immunotherapies being developed in his lab at this moment. There is lots of fascinating information packed throughout this interview, and I really enjoyed talking with Dr. McNagny. If you guys enjoyed the interview as well, consider following this podcast to stay up to date with more from this highly active field. And without further ado, here is my conversation with Dr. McNagny. So, for our listeners who might be new to hemopoietic stem cell biology, could you start by explaining in like a few simple terms what sets innate lymphoid cells apart from other cells? What are they all about?
So probably most people have heard about T cells and B cells. B cells that make antibodies, and T cells that kind of are responsible for cell-based immunity that kill the virus factories that produce the virus when you get an infection. And the reason you've probably heard of those is because that's what immunologists have been studying for well over, 50 or 60 years. And they're the target for every vaccine program in the world. Because if you get it right and you activate those cells, they develop memory and they will protect you for the rest of your life from an infectious agent. So that's why they're great cells. Problem with those cells is they're slow. And so it takes you, a week to 10 days to develop a good immune response against an infectious agent. And when you've got something like a virus that replicates, in 20 minutes, 1,000-fold, it's a foot race and you'll lose. And so that's been a mystery we'd rely on T cells and B cells, but how do they keep us alive when they're that slow? And the answer is we have some other cells that are like the T cells and B cells, but they respond rapidly and they behave like them, but they do it in a really quick way. And they start setting up that cascade that buys you the time you need to make a good immune response. And those were cells called innate lymphoid cells. They
Mhm.
were only discovered about, 10 or 15 years ago.
Wow.
And people were looking and they were finding these cells that looked just like T and B cells, but they didn't have the same markers and they weren't specific for antigens. They were more broad. And so people saw these cells and said, these are interesting. They're doing all the same job, but they don't have a specific receptor to fight an infectious agent. Maybe that's the relay switch that keeps you going before you make a good specific immune response. Yeah.
Okay. Yeah, and then is there actually a reason why innate lymphoid cells it was so recently that that they had been discovered?
Great question. So these guys come up early in development, right around the time you're born.
Mhm.
And thinking about that, right? When you come out of your mom, you've come from a sterile environment into, I'm going to call it a sewer of antigen. So you're going to the
Yeah.
world where
Completely exposed.
exactly you're naive and you're exposed to all these things all at once.
Hmm.
And so you need something that's got broad specificity and lower affinity to just keep everything generally at bay until you can start making the specific responses. So they come up early in life, they get educated, and then they colonize all of your tissues. And then after that, they just expand and contract in tissues and you don't find them in blood. And so that's probably why it took so long to discover them. They're rare and they're tissue
Hmm.
resident. And when you go to the doctor and you want to find out what's wrong with you, the easiest thing to do is take a bit of blood and look at your blood and see what's there.
Hmm.
It's the innate lymphoid cells because they're not there and they're very rare cells in the tissues. Yeah.
Okay. And you did describe innate lymphoid cells as being tissue resident. Could you maybe elaborate on why this localization is so important for their function as being
Yeah.
really rapid to respond?
Yep. So they're tissue resident. Where you find them most often is at the barrier organs. So you'll find them in the wall of the gut, where
Oh.
eating things, that's the first point of contact for some pathogens that you would eat, or on the skin. So when things infect the skin, that's another barrier organ, or in lungs, in the airways, because you're always breathing in things.
Mhm.
So they're kind of the sentinels that sit at all those interfaces you have with the external environment. And as immunologists, we kind of try to lump immune responses into three major categories. They're the ones that fight viruses, the
Right.
ones that fight bacteria, and the ones that fight parasites. And for each one of those different types of pathogen, you have a different subset of these innate lymphoid cells. And in
Oh.
order to fight those things, fighting a virus is very different from fighting a parasite. Viruses are
Mhm.
small, they replicate inside a cell. The cell explodes and it spills more virus out. And so you have to kind of protect yourself against that.
Mhm.
Parasites completely different. They're big, multicellular, they crawl through tissues. And so you need to have a very different way of responding to them. And it's these innate lymphoid cells that tell you as soon as you get that first contact, make the anti-parasite response or make the antiviral response or make the antibacterial response and don't
Mhm.
confuse the two. So that's where they kind of specialize a bit. Yeah.
Oh, and what sort of function is in charge of their specialization? So what determines the differentiation of these cells?
Good question. So I think some of the most exciting things that we're finding in my lab these days is that actually early in development, those first exposures kind of educate what the frequency is of those different subsets in your organs is.
Mhm.
And so you may have heard of these kind of microbiome. It's the bacteria that live in your gut.
Right.
And we've known for 20 years or so that while infectious disease has been declining because we've got good hygiene now, we've got antibiotics, we've got good drugs,
Right.
chronic inflammatory diseases like asthma and allergies, Crohn's disease, or inflammatory bowel disease, those have all been going up. And why, right? It's not that our genes have changed, it's
Mhm.
something about the way we get exposed has changed. And so what people found was that if you live in a super clean environment and you don't get exposed to some of those bacteria that you would normally see, back, thousands of years ago, we were crawling around in the dirt.
Mhm.
We've evolved to respond to those and to educate our immune system right after birth. If you don't get that exposure, then you'll make too many of these cells.
Mhm.
Those cells sit in the tissues and they're hyper responsive. And that's why, kids who don't breastfeed, who are cesarean birth derived, or undergo multiple doses of antibiotics very early in life, tend to be more likely to get asthma and allergies
Mhm.
and it's because they make too many of these innate lymphoid cells of the wrong type, and they think that they're in a parasite-rich environment. So they make too many of those growth factors when you get exposed to a benign thing like an allergen.
Mhm. So actually, these antibiotics that you get when you're a kid, um, that are supposed to help protect you in a certain way, they can actually come back as a sort of a double-edged sword in later life. Is that correct?
Exactly.
Hmm.
And so if whenever you've seen a little kid, what's the first thing you notice about them when they're crawling around? They put everything in their mouth,
Oh
right?
yeah.
Everything. And there's a good reason for that. It's
Hmm.
because that's the way they first get colonized with bacteria that kind of protect them. The first exposure comes as you're being born down the birth canal. You get coated with this bacteria, and right about the
Right.
time of pregnancy, um, vaginal tissue gets colonized with lactobacillus and these other bacteria that just coat the kid and they swallow
Hmm.
them. And that's
Oh.
good, right? They kind of
Hmm.
get and in fact, because we now realize that's important, in some cases, if you're a cesarean born kid, they'll kind of swab you with vaginal bacteria in order to make sure you get the right bacteria right after birth. So that's the first exposure. Then you get bacteria when you nurse. You get the
Hmm.
bacteria that's on your mom's skin when you're nestling with your mom. All of those things are good things, and getting exposed to a lot of bacteria early actually is a pretty good thing as long as it's not those really nasty ones that can invade your tissues and give you a
Right.
bad,
Yeah.
right?
Hmm.
So, So that's one big change in policies. We're telling kids, try not to keep things so clean that you don't get any bacteria when you're young. And if you're going to have a kid who gets a bad infection, of course you give them antibiotics.
Hmm.
Having to go multiple doses of antibiotics, maybe they should take a probiotic or something else shortly after the antibiotics, or maybe even at the same time,
Hmm.
so that you don't miss that exposure early in life.
Yeah, so regulating these antibiotics when they enter your body is also something that maybe researchers are just trying to figure out the right dosage, the right amounts.
Yeah, yeah.
Hmm.
And another question we're trying to investigate is, at what stage do you really first start getting exposed to these things that the microbiome produces that educate those innate lymphoid cells? And bacteria make a lot of metabolites, and some of those metabolites can actually cross the placenta. So we
Hmm.
probably also should be looking at moms who are pregnant and saying, maybe if they're on high dose antibiotics, they should be taking probiotics as well. And make sure they get exposed to the right things that maybe help educate you during fetal development too.
Hmm.
Yeah.
Yeah, so currently we're actually sort of led our conversation to the big gray area of bacteria and innate lymphoid cells. So this big gray area consists of what are some problems that we're facing? Because as you mentioned, we only recently, like within the past 10, 15 years, discovered these cells, so innate lymphoid cells. So what are some problems that or what are some technical difficulties that we're still trying to face and figure out within this field right now?
Yeah, we're trying to figure out how those bacteria make signals that educate your immune system. And so my lab's just been working on two very
Mhm.
different exposures. So we work on lung inflammation a lot. We work on things like asthma and allergies. And we also work on things that are more like hypersensitivity pneumonitis. It's like asthma, but it's a different branch of the immune system that causes inflammation of the lungs too.
Mhm.
In the case of asthma, we know that the bacteria that are missing in the kids that are going to develop allergies are these ones that make things called short chain fatty acids like butyrate and acetate and propionate. And those metabolites, if they're missing, allow a certain subset of innate lymphoid cells to expand and establish a new set point. There's more of them in your lungs than if you were exposed to the right bacteria. And not only are there more of them, they're kind of hyperactive, right?
Mhm.
They didn't go through high school well. That was the bad kids in high school that didn't get educated well, and they're hyperactive and they're doing the wrong thing at the wrong time.
Mhm.
So what we're trying to now do is say, okay, if we know these bacteria are making a metabolite, if you have to be on an antibiotic or you don't get exposed, what if we just give the metabolite to the kids
Mm.
in milk or as a supplement? Could that dampen down things so you're less likely to get allergies? So that's on the asthma side. There's a different type of asthma. There's one, I'm going to call it asthma. It's not really asthma, but it's hypersensitivity pneumonitis and also steroid resistant asthma. That's one that you may have heard of. It's one that it's really difficult to treat with the current medications. It activates a different branch of the immune system.
Mhm.
These cells are dampened down by a bile acid metabolite that the bacteria change and make into something that turns those cells down. And if you're missing those bacteria, then you're more likely to get things like steroid-resistant asthma,
Mhm.
Crohn's disease, probably things like autoimmune arthritis. Those are all driven by these pathological innate lymphoid cells that make too much of the things that cause inflammation.
Mhm.
Yeah.
Yeah. So you kind of touched on this already, but um, when innate lymphoid cells they're not functioning correctly, like what are some like problems or challenges that patients may face, and you as researchers face when you're trying to figure out why does this happen?
Yeah. So my mantra is that there is not a single disease that you and I will ever get that doesn't have an immune component to it. And you can pick things like even traumatic injury when you're in a car crash and your brain swells and you die. It's not the damage to your brain frequently that causes you to die. It's the inflammation associated with that that causes swelling in the brain and that causes the
Mhm.
neurons to check out. So, if you had a way of dampening down those pathological immune response and traumatic brain injury in chronic inflammatory diseases like asthma and Crohn's disease and allergies, um, in things like aging, aging is really an immune disease. When people like me, who are much older than you, get weak muscles and our muscles start to degenerate and turn into kind of fat and get stiff, that's fibrosis. It's your muscles, the normal tissue is starting to deteriorate and you're replacing it with fat tissue or scar tissue, really. That we think is regulated by these innate lymphoid cells too. And if you could dampen them down, you might retain healthy tissue longer and have less of that kind of scarring tissue
Mm,
up with aging. So there's just a couple of examples of kind of chronic inflammatory disease or diseases associated with aging
mm.
that you might be able to dampen down if you could better fine tune those innate lymphoid cells and tissues. Yeah.
And actually, good point. So, on this topic of trying to figure out what to fine tune and what sort of to alter in this process, is there something that your lab is doing currently that is channeling their efforts to addressing this matter?
Yeah. So we actually just published a paper looking at Crohn's disease. And we found that the major problem with people who get Crohn's disease is they get this chronic inflammation in the gut, and it leads to production of kind of scar tissue in the gut. There is no cure for that disease. It's really tough to treat. When you make that chronic scar tissue, it replaces the normal villi in your gut that, help you absorb food,
Mm,
kind of extracellular matrix and scar. And so it doesn't function anymore. And it gets really painful and it can actually block your gut. And the only
mm,
way to treat that currently is to cut a section of the gut out, stitch it back together, hope it goes away. And frequently it doesn't. So people have been trying for years to figure out what's driving that. Why do some people get it and some don't? Part of it is the microbiome. Part of it is the bacteria that you have in your gut. If they haven't educated those innate lymphoid cells well, you get too much inflammation when you get a little bit of an injury, and it just keeps going and going. And we need to find a way to turn that off. So what we showed in this paper was that the bacteria make a metabolite that activates these cells. And if we add that metabolite back, we can turn them off. Missing that it's like a dimmer switch. It's like a brake.
mm,
You don't have the right bacteria, the brake gets taken off. If we can add back that metabolite, it can act as a brake and slow that inflammation down. And then that fibrosis starts to melt away, and then healthy tissue starts to come back. And now you reestablish a homeostasis where things are working normally again.
mhm.
And most of that's driven by a loss of a bacteria that make that metabolite.
So, with patients with Crohn's disease, um, to clarify, they're missing the metabolite, which will return them back to homeostasis.
Exactly, exactly. So
Okay.
the brakes are off and they're just chronically inflamed and they don't know what to do. And if you can add the metabolite back, you can dampen those cells back down and make things more normal again. And I think that's true of a lot of the chronic inflammatory diseases.
Hmm. And when you're adding this metabolite back in, are there any like challenges within this process?
Yeah, we're trying to figure out a good way to deliver it. I mean, it looks like you could actually fix things perhaps by just adding back the right bacteria that make that metabolite. So
Mm,
sometimes you get people who get a fecal microbiome transplant, and there's
mm.
various ways of doing that, which we may not want to go into. But if you put the right bacteria back,
Mhm.
either at the back end or the front
Right?
end, and they start making the metabolite, in principle, they should fix the disease. And there's a really good example of that in the literature. I mean, I think you were interested a little bit in cancer as well, but
Mhm.
you may have heard of these things called checkpoint inhibitors. These are things that allow your immune cells to start fighting cancer cells.
Right.
And people found that some patients, you give them that checkpoint inhibitor therapy, it's got a Nobel Prize in the past 5 years, and it saves patients' lives. Some people don't respond to them, and they can't figure out why don't they respond. Some
Mm,
people, they melts their tumors away, it activates their immune response, but these other patients don't. And funnily enough, they found if they gave them a fecal microbiome transplant from a patient who responds, suddenly they start responding. So that's telling me and researchers that it really is something about metabolites that get made by your microbiome. that regulate that immune response. And I've given you pathological situations where that's bad. Here's a situation where if you can give the right microbiome back, it's actually good and will help you fight cancer.
Mhm. Yeah, and the process of adding the missing metabolites back in, have has this technology been around long enough to actually see the result? So patients have received this transplant.
It's really new. And for so far, it's been kind of correlative. So if they, for the example of Crohn's disease, they found this metabolite because people were looking at metabolites from the gut in patients who didn't have Crohn's disease, and they found something was missing in the patients that had Crohn's disease. And then they realized that if they put that on lymphocytes in a dish, it turned them off. So
Mm,
that was the correlative data. What we then did is we said, hey, we've got a really good mouse model. This is not ready for treating the patients yet, but we can try it in mice. We started adding that to the mice that we were inducing to have Crohn's disease, and it flatlined them, so they were doing great. So that's the first step in showing that it actually could be useful in treating patients. And I'm sure there are clinical trials going on now to find out if the same metabolite will work for Crohn's disease.
mhm,
It's one of those things where, you make a discovery, you have an idea, you first show it's working in mice, then you have
mhm,
to get the ethical approval to try it and show it's non-toxic in patients. Then you have to get a good clinical trial showing that now that it's non-toxic, now we can test on a few patients who really are desperate need of the treatment. And then if it shows some promise there, then you do a bigger trial and eventually it becomes standard practice.
mhm. So it's a very, very lengthy process. And and because research never ends, like I don't personally don't believe that research ever ends and we never find a true answer for anything. At this point of your research about Crohn's disease and then the ways to treat it, like are there any future advancements that are coming in right now that are being implemented or undergoing more research?
I think there's a ton. So, we kind of started off saying that I think every disease has an immune component and it makes it
Hmm.
better or worse. And it's only a matter of understanding which cells are driving that bad response. And once you understand that for a specific disease, because it's going to be different for every disease, then all you have to do is design a therapeutic based on knowing what the target is. All right.
Hmm.
There has never been a better time in science for doing that. We've gotten so many great tools now that really it's just a matter of a little bit of time and a little bit of money, I think, to treat any disease. We're much better than we ever used to be. The problem is having the money and the funding to do it. And I think we do really well in Canada on a very limited amount of money. And I think if there was more money in the system, we could do a lot better. The States has always had money. Currently, they're undergoing some problems, as you may have
Yes.
heard, budget cuts for research. That's going to be devastating. And to my mind, that might be an opportunity for Canada. If they're kind of quieting things down on the research side in the States, why don't we take it up and become the next leaders in research? I think we well could. We've always fought above our weight class in Canada. We've always had great science on a smaller budget. I think with a little more budget, we could do amazing things. So that's my pitch for more money for science.
A podcast is a great way, I think, to sort of get your opinions out there.
It's a fascinating business to get into if you're just a curious person, but if you want to have an impact on a world stage, I think we really could do that in Canada.
And I think that's um, a good thing to know when you're, as me, someone who's aspiring to become a biomedical researcher in the future. I think that will
Yeah.
lift a lot of young people's spirits.
It's a fantastic career. All you need is a little bit of curiosity and an eagerness to learn something. If you like solving puzzles, then this is the most wonderful job in the world. And I always tell my students, look, sometimes times are tough, sometimes our times are good. As long as you have the longevity and the dedication to weather those tough times and then enjoy the good times, you'll be a successful scientist. It's like art. If you really, really enjoy it and you're passionate about it and you invest in it, you'll find a way to make a living and you will never, guaranteed you will never be bored. There's always a new question. Yeah.
Mhm. Yeah. And just bringing everything back to all this new technology, all these advancements that you've just been talking about how as time goes by, we've gained more experience, new tools to sort of figure out this huge realm of biomedical science. How might our understanding of innate lymphoid cells change our overall view of the immune system in the coming years, so in the future?
I think there's so much there. I think they're really the drivers of almost any chronic inflammatory disease, and it's just figuring out which ones are activated and which ones are repressed to determine how to fix it. I've got students now who are trying to find ways to grow them from human and alter them in a way that could benefit patients. So we're finding ways to grow them as a cell-based therapy. Much simpler than that is to find the molecular switches you use for turning them on and off. Can I find a growth factor that turns them on when I need to? Or if they're the bad actors in a disease, to find something that blocks the growth factor they need to just turn them down a little bit. So, as I was pointing out, that kind of high throughput screening and ways to find ways of turning things on and off, it's gotten actually pretty easy now if you've got the money to do it. We've got single cell sequencing. These were things that were mind blowing when I was a graduate student. You can take a tissue, with 100 different cell types, and within a matter of a week, know every single cell that's in that tissue, know what they're making, and see how that differs in a patient with disease and one that doesn't. That makes it pretty easy to figure out what's going wrong. And, with those kind of tools, now that you know what's going wrong, you just have to find a way to turn that thing that's going wrong off. So that's
Mhm.
where I think the excitement is. You're going to see that in chronic inflammatory disease. You're going to see it in aging diseases as well, and hopefully in ways that you could treat things like autoimmunity too. So be on the lookout. There's going to be a tidal wave of science if we keep investing
Mhm.
in improving healthcare.
Yeah, and if we could actually, on the topic of going into the future and then what that may bring, if we could just fast forward, let's say 20, 30 years and envision like the dream innate lymphoid cell based therapy for a patient, what do you think it would look like?
Oh God. Yeah, I think simple things like turning them on and off with just something you take as a pill, would be
Mhm.
amazing. We're working a little bit on that. So, and this is a little bit of a deviation, but to me, it's mind blowing that we had a brand new disease that came out, the SARS-CoV-19 virus that threatened the entire planet. And in a matter of 6 months, we had a vaccine that for a few pennies you could give to billions of people, literally billions of people around the globe, and save their lives, prevent them from getting that disease. Right? That's mind blowing. What if we could use that same technology that people use for that new vaccine to activate innate lymphoid cells? Maybe a certain subtype that's causing this disease or that disease, and we could do it for pennies. Can you imagine how much that would improve healthcare, right? People who are normally waiting for these really super expensive immunotherapies that cancer patients get that cost hundreds of thousands of dollars. If now we could manufacture these things that would allow you to do that for just a few pennies, my goodness, that would really change the way that we treat people, keep them from suffering.
Yeah, that's an incredible thought, honestly. And I know there are so many people around the world that their lives and the people that they love are affected by these diseases. And it's just really great to know that maybe it's just almost within reach, possibly. And on that note, for all those similarly aspiring biomedical sciences who are maybe tuning in, is there anything like a motto or something or a small token of advice that you would give them right now when they're about to get involved in this field?
I always tell my students that are just joining my lab, science is like so complex that they tend to go into the lab thinking I'm going to do this experiment and I will solve this problem. And what happens remarkably is 90% of the time you go in and you look at something and it doesn't give you the answer you expected. It goes in a completely different direction. And some students will throw that away and say, oh my God, it didn't come out the way I did. I have to do it again. Other students will look at that and say, wait a minute, that's not what I predicted. That's really cool. That's different. And it may not solve the problem I was looking at here, but I can see how it could easily solve a completely different problem. It's having that pragmatism to capitalize on the unexpected that really makes science so interesting. And it also it distinguishes the people who are going to do really well in science from the ones who are going to just kind of do the stamp collecting and follow the next, minor increment. So as long as you're curious and you like solving problems and puzzles and something unexpected comes up and you get excited about that, you're going to do great in science. Yeah. That would
That's
be my motto.
yeah.
Be curious and be excited about the unexpected results.
Yeah, I think that's a really good that's a really good motto. And it's really important too because you don't just stop when you find like a challenge or an obstacle or something that just doesn't go right. Like not at all. You have to realize that everything is interconnected somehow. And if this doesn't solve one thing, what can it lead to another thing if I continue to pursue it? Like there are
Yeah,
no mistakes, I don't think.
absolutely right. I have a student who is doing a comprehensive exam, and she was asked, well, you're trying to cure cancer with these cells that you're making. What if that doesn't work? What if they actually drive cancer? And they said, well, something that activates cancer cells will be useful in a completely different disease where you've got, no immune response. Well, now we want to drive an immune response. Well, let's do that. We'll find a way to use the same tools for curing a different disease. So just don't throw anything away. Realize that it could be used for something else. And then the last one I wanted to just touch on for anybody who might be listening about the vaccine stuff. And you're going to hear a lot about that. And people are vaccine skeptics or fearful of vaccines. And it's remarkable to me, the same people who might get cancer or have a loved one who gets cancer and will be begging doctors for access to the latest therapy are the same people who tend to be afraid of something like a new vaccine that can save their lives and is really simple. And so I just want to encourage people to maybe trust the scientists because it's the same people, developing your cancer therapy as developing the vaccine therapies that are stripped down and super simple and super safe, much safer than anything we've had in the past. Just, trust the scientists a little bit. We'll take the time to explain it and try our best. But give us a little bit of trust. We're not out there to, create something that's going to take over the world. We're out there trying to help people. That's really at the heart of most scientists that I know.
Yeah, and that's also as well sort of the researcher that I'm hoping to be, and I know that you are. So yeah, thank you so much for what you do and all you researchers in the world. They're really inspiring the next generation.
Delighted. So listen, next time you want to do another one, I can do another one with you, and I can tell you about. We've got a cancer immunotherapy that we're trying to develop, and that's more along the hematopoietic stem cell ones. And I'll just give you a teaser: is that I worked for many, many years on these really rare hematopoietic stem cells that you transplant into patients and you reconstitute all of their blood cells, right? And we were working on the stem cell protein. I was trying to figure out what it did on stem cells, and I figured out it makes them more mobile and kind of invasive. And then we realized, well, cancer is a mobile and invasive disease. I wonder if any of these proteins that help stem cells kind of engraft ever get turned up in cancer. And we found this one of them is turned up on virtually every subtype of cancer as it becomes metastatic. And wow, that's great. Well, now we can predict the patients who will and won't die of cancer because we can pick out those metastatic ones and we can separate them from the ones who are going to do fine with just a little bit of surgery. And then we thought, okay, if it picks out the bad ones, what if we try to turn it off? Does it save people? And at least in the mouse models, turns the cancer completely off. If you get rid of that protein, the tumors stop going. And so now we're making antibodies that we're hoping to take into the clinic to try to target that tumor stem cell protein, that protein that helps tumor cells metastasize. So I can give you that as a teaser for another podcast if you want to
Wow,
do one.
that's really cool. Wow. I'm just starting to realize how really like how grand research is, and especially even like even just one topic like hemophoetic stem cell biology, even in this subfield of like biomedical research, there's so much going on. There's so much potential in it. So that's
Absolutely.
really enlightening. If if maybe later perhaps I can reach out to you regarding cancer and then what you guys are doing in the lab about it and just host another interview, that would be awesome if you're willing to do that.
Happy to show you a few slides of the kind of schematics of how we think things work too, if you like.
Yeah, that's awesome. And I hope future listeners will also enjoy that as well.
Fantastic.
Yeah, thank you so much.
Very good.
All right. Thank you so much,
Bye-bye.
Kelly.